Awesome. Yeah, thank you so much. So I guess maybe I'll do a brief info. So Dr. Anna Carter is a professor in the UCSD Department of Medicine. And her research, she does a lot of really interesting stuff with cancer genomics. And there's this really interesting mixture of kind of the computing side of it with machine learning and all sorts of kind of modern computer science techniques, but then very closely tied to the biomedical application. So yeah, thank you so much for joining us, Dr. Carter.
Sure. Nima, thanks for having me. I'm gonna sort of hide my sidebar so that I can see my slides. So I guess feel free to, I guess, interrupt me if there's any questions or anything. So yeah, I was going to maybe start with a little bit of background on how I got where I am, and then give a little primer on genetic variation in cancer, and then talk a little bit about some of our latest work, which is focused on looking at genetic background and its role in anti-tumor immunity and how different people's immune system interacts with the tumor. So my parents are actually artists. That's my dad there. He's an amazing painter. And my mom has always done things to complement what he does. Like here she's doing, creating a gold leaf frame. And so I wasn't really, I didn't grow up in a, a family of professors or engineers or anything like that. But still, I found that I was really interested in how things work, and tended to, take over problem solving and electronics and wiring things up in the house. And so eventually, I encountered, actually, luckily in high school, an electrical engineer who got me interested in engineering. And then I went to, this is Speed School, the engineering school at the University of Louisville, in the town in which I grew up. So I ended up doing a 5-year master's program studying electrical engineering. And then this program actually involved a number of internship experiences where you could go work at different places and see what engineering was like in the real world. So I ended up at places like RCA, programming infrared remotes, and working on the last generation of these cathode ray tube driven flat screen TVs. This was an 80-inch one. I don't think you could find anything like this anymore. We've all moved to flat screens, which are much nicer. And then I also did an internship at Lexmark working on problems in the printing space. But I actually found that working in this area didn't really get me very excited about, what I did day to day. And a lot of the people at these companies that I respected were leaving to get PhDs. And that's why I decided I would look into graduate school. I ended up going to Johns Hopkins to study biomedical engineering for my PhD. And coming from an electrical engineering background, I thought I was going to do something with neuroprostheses, like translating electric signals from the skin into motions of artificial limbs and things like that. There's some really cool work being done there. But interestingly, there were a couple of things that changed my direction. One was the guy who ran this lab apparently he didn't have a very good reputation as a good advisor. And the other was that when I went here, I had the opportunity to learn my biology through taking the first year of medical school. And when I did that, I became completely fascinated with how cells process information and how genetic mutations in the DNA can change cell behaviors and ultimately result in diseases. And so I sort of did a 90-degree turn and started studying genetics with computation. I ended up joining the lab of Rachel Karchin, who was a new faculty at Hopkins at the time, and she was using machine learning to try to predict the effects of DNA mutations that altered protein function. So this funny schematic here is a 3D structure of a protein where the backbone is rendered as a ribbon. You can see there's like these cool helices that it forms, and some other loops and turns and things. And the function of a protein is determined by its structure. And when you have an amino acid substitution, like this valine 600 substitution here, it can change the structure, which can end up changing the function of that protein. So I started working on machine learning methods to try to predict what the functional consequences of mutating a position in a protein would be on that protein's function. And then we ran into a new problem emerging in the field. So this is Bert Vogelstein, Ken Kinsler, and Victor Velculescu, and they are also at Johns Hopkins and were at that time just getting the results back of the first whole exome sequencing studies of tumors. And by whole exome, I mean all the regions of DNA in the human genome that encode proteins. And they were finding a lot more mutations than they thought were necessary for cancer to develop, which raised a new challenge, which is which of these mutations that we see is causing cancer and which ones are just coincidentally also happening in the cancer cells. This is called figuring out which mutations are drivers versus which are passengers. So this ended up becoming my thesis project, working on a machine learning tool to do that. It culminated in a few different software tools. CHASM is the cancer-specific high-throughput annotation of somatic mutations designed to classify drivers versus passengers. Uses a random forest classifier, which basically is a collection of decision trees where each decision tree gets to vote on whether a mutation is a driver or a passenger, and then the votes are aggregated across the forest. It actually is available still via this Cravat web server where you can input variants. I also developed another tool, VEST, which is not for cancer, but for just genetic mutations in someone's inherited genome. And I'll try to explain how those types of mutations are different a little bit later. But after my PhD, I found that I still wasn't satisfied. This tool does a decent job trying to predict drivers versus passengers in a given protein based on these local features of the protein, but proteins don't exist in isolation. The inside of the cell is this mix of all these different proteins that are sort of wobbling around and running into each other, and information is transferred through these interactions. Sometimes you'll see people try to depict this with pathways, which can get quite complex. So here you're seeing on the top the cell membrane, and the middle here is sort of the nucleus, and information is being transferred from the membrane to the nucleus where you can regulate gene expression and activities of the cell. But you can see that there's a lot of different proteins and signaling molecules inside the cell that have to work together in some kind of coordinated fashion to get that information where it needs to go. And so you can imagine that mutations in some parts of this network might be much more important than mutations in other parts of this network. So probably the context of the specific protein is going to be really important to take into consideration. So I joined Trey Idicker's lab here at UCSD, and he does computational systems biology, studying networks of proteins, interacting proteins in health and disease. And so the idea was to try to integrate sort of these protein level features, predicting amino acid substitutions, with now network level features to better understand the potential of mutations to be drivers versus passengers in cancer. And actually, while I was doing my postdoc in Trey's lab, I also applied for a grant called the NIH Director's Early Independence Award, which I got, and that subsequently led to me becoming an assistant professor at UCSD, getting space, and then ultimately this work wound up being part of that grant and one of my first publications after I transitioned to a faculty position. So I think that was kind of my journey. I was just different opportunities arose at different times, and I followed what was exciting and interesting, and it has led me here. So now I have this lab at UCSD, and we're interested in studying genetic variation, not just thinking about the protein structure and function, but trying to think about it at multiple scales, including protein interactions, signaling pathways, networks, et cetera. And we're particularly interested in precision medicine and understanding how these mutations are relevant to diseases and especially cancer. Yeah, and we use, various computational tools, whatever's appropriate to study these variants and answer specific questions that we have. So now I'm going to switch into talking about cancer specifically. So I don't know how familiar everyone is with cancer as a disease, but cancer is known to be a genetic disease, and I guess some of the classical examples have been cancer syndromes. There's a small number of these syndromes where people have a really high predisposition for developing cancer in their lifetime, because they have a mutation in a very important gene. And these are usually genes that are involved in DNA damage repair. If you have a defect in one of these genes, then you will accumulate mutations in your genome much faster, and ultimately the accumulation of mutations greatly increases your risk of developing cancer. And you may have heard some examples like Lynch syndrome or folks with BRCA mutations who, maybe made a radical decision to get bilateral mastectomies to prevent development of breast cancer, et cetera. These, however, account for a relatively small fraction of cancers, 5 to 10%. The rest could be considered sporadic cancers and are often associated with exposure to mutagens, things that cause mutations to accumulate in your genome. For example, UV radiation or cigarette smoke. However, even though we think these are quote unquote sporadic, we still see that there is this statistically unexpected rate of these similar cancers arising in families, which suggests that there's more to the genetic risk of cancer than just these rare high penetrance mutations that account for cancer syndromes. And there has been a lot of work trying to understand the genetic predisposition to various cancers. There's also non-familial cancers. Sometimes mutations accumulate because of things like UV radiation or cigarette smoke, and you just get unlucky enough to get the right set of mutations to cause a cancer. In terms of the familial risk, there's been some interesting studies of twins where they, asked if one twin got cancer, how likely was the other twin to get cancer? And then they use that to estimate how much genetic heritability is accounting for this familial risk. And so you can see sort of the ranking of tumor types based on the ones that seem to be most due to shared genetics, with melanoma and prostate being at the top and then the list decreasing, but something like 33% of cancer is thought to be attributable to heritable factors based on these types of analyses. And so here to study how germline or inherited genetic background might contribute to cancer risk, they've performed a number of what are called genome-wide association studies. The idea being you get a large group of people with a disease, a large group of people without the disease, and then you study these SNPs. That stands for single nucleotide polymorphisms, but these are basically markers across the genome. And the markers that correlate with the occurrence of disease, so the markers that are more prevalent in your high-risk population versus your healthy population, indicate that something in that region of the genome is probably causing the increased risk of cancer. And then there's a lot of work downstream of that to try to figure out what each of these associations, here you're seeing all the cancer associations distributed across the human genome marked in purple. And so there's a lot of work to be done to figure out what at that location in the genome might be accounting for the risk. So that's the genetic contribution, like the inherited genetic contribution to cancer risk and why it's thought of as a genetic disease. But the other part of it is that when we study tumors, we can see that they also accumulate mutations, and they seem to cancer seems to arise only when you accumulate a set of really bad mutations. So this is actually a model that was developed by Bert Vogelstein and is referred to as a Vogelgram from his studies of colorectal cancer. And here you can see sort of the progress. This is, I guess, birth, and somewhere around 34 years of age, this person had a cell that acquired a mutation in APC and beta-catenin, and it just gave this cell a little bit of a growth advantage. The cells grew a little faster. And then one of those cells accumulated a mutation in CDC and maybe some chromosomal instability and started to grow a bit faster, and so on and so forth. And so the cells become progressively faster growing, more persistent in their survival, and eventually become aggressive and invasive and create an actual tumor. But the timeline for this can be over many years. And usually we're diagnosing the disease somewhere toward the end here when it's already very dangerous. And you may hear a lot of research geared toward trying to detect these diseases when they're much earlier, more close to their onset, so that we could prevent people developing the life-threatening cancers. So anyway, conceptually, it's the accumulation of these nasty driver mutations that allow this otherwise normal cell to suddenly become disease. So how do we determine what those somatic changes are that the tumor is acquiring? We do that with DNA sequencing. So usually what will happen is you'll sequence the tumor DNA and then some like blood matched normal DNA from the same individual. You throw this on a sequencer, you get a whole bunch of reads that tell you what the DNA sequence was. These have to be aligned back to a reference genome, so we figure out the address in the genome where the read came from, and then we can compare it to the reference to see if there's a difference or not. Was there a mutation? We have to do this for both the tumor genome and the germline genome because everybody is a little bit different. Any two of us standing next to each other differs easily at 3 million different sites in our genome, but the tumor may only contain, 20 relevant mutations. So we need to make sure we account for the individual difference from the reference so that we don't accidentally include those as somatic mutations in the tumor. So we can rule out all the mutations that are differences from reference, and then we see what's left over using a statistical comparison. Some popular methods are Mutect, eBayes, and Strelka. And so then you get out a list of confident DNA mutations that were specifically acquired in the tumor DNA and not in the background of the individual. The problem becomes, though, that there's a lot of mutations. So this is a public study funded by the NIH called the Cancer Genome Atlas. They sequenced hundreds of tumors from all these different tumor types. This is uterine, melanoma, colorectal, lung adenocarcinoma, stomach cancer, et cetera. It's hundreds of these tumors. And then if you look across these tumors, you see that they found many, many mutations. And this is sort of breaking the mutations down into different categories. And here we're just looking at protein coding mutations. And then if you divide the number of mutations found by the number of samples, you can also notice that some tumors harbor fewer mutations and other harbor more. This uterine cancer here often has what's called microsatellite instability, which is a form of DNA damage, repair, malfunction. So they accumulate a lot of mutations. Melanoma, we usually see UV radiation contributing to mutagenesis. Colorectal cancer, again, that microsatellite instability, lung cancer, smoking, and so on and so forth. And so you can see that these processes or these exposures can introduce a huge number of variants into the tumor genomes, making it much harder to track down which of these is actually a causal driver mutation. And again, we think probably somewhere on the order of like 3 to 8 mutations is all that's required to transform a cell to be cancerous. So we've learned a few things by studying tumor genomes that can help with this a little bit. First, even though every tumor harbors a different set of mutations, we recognize tumors as tumors because they behave in specific ways. The cells resist cell death. They don't die appropriately. They start generating their own proliferative signal. So they're independent of external signals. They can just grow on their own. They deregulate their energetics, they accumulate mutations, they evade the immune system, and so on and so forth. So that all tumors have in common. And ultimately, what we found is that the subset of mutations that are drivers are the mutations that enable the tumors to accumulate these characteristics, and they tend to fall in a set of key pathways that regulate cell survival, cell fate. And by cell fate, we mean cells differentiate to a final functional form. And loss of that differentiation makes them dysfunctional and increases the risk of cancer. And then the genome maintenance, which is frequently lost in these tumors. So you can almost think of it, this is the analogy presented by Han and Weinberg here, as a subway map where each of these are these different pathways, molecular pathways regulating things like apoptosis or cell cycle and replication and things like that. And there's many different proteins that are involved in activating or inactivating the activity of these pathways. So you just have to get mutations in a couple of these proteins in the right combination for the cell to become cancerous. And so knowing what these pathways are and which genes participate in them is really helpful for trying to fish out which ones are the driver mutations. Although it still remains sort of an open research area, we've identified a number of cancer genes and mutations, but it's not always trivial for any one tumor to understand what are the mutations that are driving or causing that specific tumor. So that's what we want to do in precision medicine, ideally, right? We want to identify individuals at risk for cancer. The best is if we can just use genetic background and find people early on that are high risk so that we can to sort of screen them aggressively to try to detect their tumor early so we can actually take an action and prevent them from getting disease. But if someone does get a cancer diagnosis, we sequence the tumor and we look for the mutations that might be causing it, because some of these mutations are also an indicator that that tumor is going to be vulnerable to a specific drug. So then we can use the tumor genome to match individuals to the best drug for their disease to try to shrink or eliminate their tumor. And then where we are now in my lab is thinking about how genetic background might also shape the evolution of the tumor and its acquisition of different somatic mutations. Every tumor is evolving on the genetic background of the host. And so the mutations that are going to optimize the growth and survival of the tumor might be very patient specific. And that might explain why we see a lot of differences across individuals on whether they are going to respond to particular cancer drugs. You don't want to, if you can avoid it, assign someone a drug that is not going to benefit them, but might have uncomfortable or life-threatening side effects. Okay, I am going to start talking about our more recent research now, but I can pause for a sec. Does anyone have any questions about cancer or the accumulation of mutations?
Any questions, folks? Feel free to type in the chat. Feel free to unmute. No questions so far?
Okay,
Perfect.
all right, I'll dive in. So in this last section, we're going to talk about studying genetic background as a determinant of anti-tumor immunity. And there's a lot of interest in studying the immune system in cancer. As you saw before, tumors learn to avoid the immune system almost universally. It's a hallmark of cancer. So this suggests that an individual's immune system is placing pressure on the tumor. It has to be able to detect it and threaten it for the tumor to have to evolve mechanisms to avoid it. And we found very recently, and this was the breakthrough of the year in 2013, that if you can reverse the mechanism by which the tumor has avoided the immune system, then immune cells can come in and kill the tumor. And this is called immunotherapy. We've seen a huge increase in the number of different immunotherapy drugs in the last few years, and immunotherapy, especially checkpoint blockade, which I'll get to in a few minutes, has become a standard of care for several different cancer types. However, not everyone responds, and so it's important to understand what is determining an individual's response. And we set out to try to understand whether differences in the inherited genome of individuals pertaining to their immune system might be contributing somewhat to this variability in response. So anti-cancer immunity is somewhat complex. I'm going to focus on certain aspects of it and give an overview. I think the key thing to understand is there's a quality control mechanism in your body in every cell. It's called antigen presentation. And basically what happens is there's this molecule that all of your cells express called the major histocompatibility complex. Its job is to present peptides derived from chewed up proteins inside the cell on the cell surface so that your immune cells, in this case, this is a killer CD8 T cell, can come along and just check, is this cell healthy? And there's a process by which these killer T cells are pruned so that you remove any that recognize proteins that are normal. Otherwise, you would have autoimmune disease. So your T cell repertoire should be entirely comprising cells carrying receptors that would not recognize self, but should be able to recognize something foreign. So if you got an infection, the infected cell could be detected and eliminated. Now in cancer, you can accumulate these mutations that affect protein sequences. So if a protein harboring a cancer mutation is chewed up into peptides and the mutant peptide happens to bind to the MHC and get displayed on the cell surface for T cells, the T cell might recognize this as foreign and attack the cancer cell. This is the recognition of antigen presented by MHC by T cell receptors is part of multiple different immunotherapy strategies, including neoantigen vaccines, immune checkpoint blockade, and adoptive T cell therapies, because they all require T cells to identify a target. And so this is a really important molecule, the MHC, for immune surveillance. And because you have to have peptide presented by MHC for immune surveillance, we decided to start looking at individual genetics at that level of the MHC. And there's actually a lot of differences across individuals in the MHC. So to give a primer, of course, it's a bit more complicated than what I just went over. There's two classes of MHC. Class 1, which is expressed by all nucleated cells, presents peptides from inside the cell, like I just described, to these killer or cytotoxic CD8 T cells. But there's this other class of MHC called class 2, which is expressed by antigen presenting cells. So these are professional immune cells that just go around scooping up proteins and things from their environment and displaying those to a different class of T cells called helper T cells. And the concerted effort of helper T cells and the CD8 T cells is required to get a sustained immune response. The MHC molecules are all encoded on the short arm of chromosome 6 in a region called the human leukocyte antigen region. And the MHC1 is encoded by the HLA-A, B, and C genes. Class 2 is a bit more complicated. But in human populations, there are thousands of different versions of these genes. And that's because they're in an arms race against pathogens. Pathogens try to mutate and evolve to avoid detection by MHC. And MHC has to also encode a great deal of diversity in order to capture as many pathogens and make sure that we can mount an immune response. So, like I said, you still only get 2 copies, one maternal and one paternal, each of these 3 genes, but you're sampling those from this large pool of possible alleles. These will all have different sequences, and if the sequence has effects on the structure of the peptide binding groove of the MHC, it could change which peptides are bound and presented by this molecule. So you can imagine that different people have different alleles, might be able to present different peptides from their genome or their proteins on the cell surface for the immune system. So we thought this might be important for cancer, and we studied how the inherited genotype of individuals interacts with the set of mutations in their tumor. So we looked if their MHC could present mutated peptides in their tumor, and then we asked whether it was important for immune responses. And this is an example in the context of an immunotherapy treatment. So I thought I had a slide on immune checkpoint blockade in here. I'm going to go back here for a second and try to explain it. So in this middle panel right here, you have the MHC presenting a mutation, and here's the T cell receptor. There are these secondary co-stimulatory and co-inhibitory interactions that can happen. In this case, we're seeing PDL1 and PD1. PD1 is a co-inhibitory receptor on the cell surface of T cells, and if it encounters anti-PD1 on if it encounters PDL1 on its target molecule, it will recognize it as self and shut down the immune response. And what these antibodies here are these immunotherapies called checkpoint inhibitors, where the antibody is specifically designed to bind either to PD-L1 or PD-1 and block their interaction with each other. That way, when the T cell receptor binds to the peptide MHC, this secondary interaction can't happen, and you can't suppress the T cell response against the tumor cell. That's the idea behind checkpoint blockade. So here we're looking at a group of 77 tumors that were treated with these immune checkpoint blockades. One of the things that's been reported in the literature is that having more mutations in the tumor is associated with better response to these immune checkpoint blockers. So blocking the interaction between PD-L1 and PD-1 on T cells and tumor cells only helps if there are mutations that can be presented by the MHC to drive this kind of immune response. And if you have a lot of mutations, there's a higher chance that you're going to have one that's presented. So this is what you see in this is called a Kaplan-Meier plot. This is how long people survive post-treatment with their tumor. And you can see that the tumors that had a high mutation burden, they had a much longer survival after treatment with immunotherapy versus those treated with immunotherapy with a low tumor mutation burden. And this has been seen multiple times in different trials for several different tumor types. What we did is we calculated a score that basically integrates each of the HLA A, B, and C alleles that a person carries versus the affinity of the MHC encoded by each of those for mutations that were found in their tumor. And we asked, what happens if we say not only do they have to have a lot of mutations, but one or more of those mutations has to be really well presented by an HLA allele that they carry, so by their MHC. And what we found was you could split the high tumor mutation burden group into two, where these folks in blue both had a high tumor mutation burden and had one or more mutations that would be really well presented on the cell surface by their MHC. And they did the best. But if you had a high tumor mutation burden and none of your alleles could present effectively the mutations in your tumor, those patients did much worse. So it seems here that your genetic background in terms of which of these HLA alleles that an individual inherits can interact with the mutational landscape of the tumor. And if mutations are well presented, then the immune system can recognize the tumor and will respond better to immunotherapy. And if not, then immunotherapy can't really generate an effective response in those individuals. But the MHC is only one protein complex involved in antigen presentation and immune responses to tumors. Overall, it's much more complicated. Here I've tried to just give an example of a couple of different pathways. This is the class 1 antigen processing and presenting pathway we saw it a minute ago. This is class 2, uses slightly different proteins. Then we have these, as I said, co-stimulatory and co-inhibitory molecules like PD-1 and anti-PD-1, or sorry, PD-1 and PD-L1, and then CTLA-4, which binds to CD80, et cetera. So you want sort of the activation signals to be present and the inactivating signals to be absent to get an effective immune response. And then T cell receptor signaling to activate the T cell is also highly complex and involves a number of different proteins. So we don't want to stop just at looking at genetic background at the MHC. Genetic background could affect the sequence or the expression of any of these different proteins that might be very important for immune responses. So to undertake this, here's just examples of very many publications that show the influence of genetic background on immune responses in humans. So it's reasonable to think that inherited immunity is going to also be important for tumors. But so this really talented MD PhD student, Megan Apagadala, undertook this project a couple of years ago, and she collected a bunch of measurements from the TCGA data set. They had RNA sequencing of the tumor, which would also happen to sequence immune cells that were present inside the tumor at the time it was resected and then sequenced. And so she identified a bunch of different immune markers of interest and got their expression across nearly 10,000 tumors in the TCGA. You can see that there's some correlated expression due to high levels of infiltrating lymphocytes. These are the T cells or macrophages, another type of immune cell, et cetera. But there's a lot of different kind of functions encoded as well. Not all of these measurements are correlated with each other. The next she asked is, can I detect any evidence that genetic background is accounting for variability in the levels of these markers? And she found evidence through the statistical analysis that 157 of these different molecular characteristics related to immunity had were being influenced by the genetic background of the individual. This is basically the proportion of variation that is due to genetics out of the total variation in the expression of these various genes. And you can see there's a few, especially in the HLA region, that genetic variation is strongly associated with expression. And then it's more subtle with others. But finding that we can explain it with genetic background, then we can go and perform one of these GWAS studies to try to figure out what in the genome might be regulating the levels of these in an individual, explaining the sort of genetic background and how it shapes the expression of these immune genes. And most of these are local SNPs. It's a change in the DNA sequence, usually outside of the gene in a regulatory region that just modifies how strongly it's expressed. So we found a whole bunch of these. Oh, that's where my slides went. Sorry about that. We found about 800 of these, and we wanted to ask then if they would be important to anti-tumor immunity in cancer. And I'm skipping over a few parts of this just because there's a lot, but we found a number of these SNPs associated just with general predisposition to cancer. Some were associated with how aggressively the disease was progressing. But what we're most excited about is that we were able to find some that were important for response to immunotherapy. We collected a number of data sets where a cancer had been treated with immunotherapy. We then tried to back these germline variants in there. We could only back in about 525. And then we studied across the melanoma cohorts if the SNPs were associated with response. And while nothing was a real standout, we did see a number of SNPs that looked a little bit interesting. And when we looked at the cumulative burden of these, if you had multiple of these SNPs that were associated with response, you were much likely to be a responder to immunotherapy. Based on this, we built a machine learning model called an XGBoost model, similar to a random forest with a couple of other cool additions. And we trained on these 4 melanoma cohorts to try to predict, based on these germline variants, whether someone was going to respond to immunotherapy or not. And then we applied our model to these other 2 cohorts to see if it would work. And here you can see, when we look at our score predicted by the machine learning model, that we give much higher scores to the folks that are responding both in kidney cancer and in non-small cell lung cancer. So this genetic background is important in melanoma and kidney cancer and lung cancer for determining whether someone is going to respond to immunotherapy or not. This is another way to show the same data, called an ROC curve. One of the cool things that we can do with these XGBoost models is we can go back and see what features were the most informative for their prediction. So this is the top most informative SNPs. These are just SNP identifiers, but these SNPs were all identified in a GWAS with the expression of some molecular characteristics. So here we have a SNP associated with infiltration levels of T follicular helper cells, here dihydrofolate reductase expression, et cetera. So these SNPs now tell us that variation, genetic variation in the expression of these genes is also associated with response to immunotherapy. And we can now try to see if these genes are actually causal. In other words, are these genes contributing in some way to remodeling of the immune microenvironment to make tumors more or less responsive? And so if we can do an experiment now. Here we've got a colorectal cancer model in mice, and we take this gene, it's one of our genes implicated by our SNP, by our XGBoost model, that happens to also have an inhibitor available. And I'll note that higher expression of CTSS was associated with poor response to immunotherapy in our model. So it makes sense, if we knock this down, we should get better response. So we used a CTSS inhibitor that was available, and we compared to that antibody, anti-PD-1, that blocks the interaction between the inhibitory interaction between the tumor and the T cell. and the combination of the two relative to mice that were untreated. And you can see here, this is the growth rate of the tumor when they were untreated. Both the anti-PD-1 and CTSS small molecule inhibitor result in slower growth of the tumor, and the combination of the two results in even slower growth and prolonged survival. This is the Kaplan-Meier curve for this really aggressive tumor in these mice. And then when we studied the immune cells in the microenvironment, we actually saw a shift when we used the CTSS inhibitor away from a suppressive macrophage signature towards a more inflammatory signature that's conducive to a stronger intratumor immune infiltration and response. So we thought it was really cool that the SNPs we identified could also point us to genes that would be important for immunotherapy response. Okay, so I'm going to just wrap up here, and hopefully you've learned a little bit of something about how genetics contributes to cancer risk and cancer progression, that the inherited genome is influential in host anti-tumor immunity, and variability across individuals is also helping to explain why people respond better to these therapies than others. And we're very excited that we can study these polymorphic variants in the context of what we call selection-driven molecular characteristics, or the hallmarks of cancer, to try to identify potential drug targets in the case of, for example, CTSS, to remodel the immune microenvironment in a way that will make it more receptive or sensitive to immunotherapies. And then before we end, I just want to say that this type of work would not be possible without an amazing team of fantastic graduate students who you can see here, and many fantastic collaborators as well, and some funding sources. All right, I think I can stick around for a couple of questions if there are any.